I'd like to describe for you a historical prospective study. And oftentimes, what makes it confusing is the word prospective because there, as you know, there is something as a prospective study, which I'll cover next. But this is a historical prospective study, and it's usually distinguished between uh, itself and a case control study. So I want you to begin to think about that as we go along, and then next I'll cover I'll cover the prospective study and talk about a couple of study designs. Um, a historical prospective study, obviously an observational study, that's all we've been addressing here for several slides now, is also known as a cohort study, a historical cohort study, a non-concurrent prospective study, and other terms. And I was trying to tell you that oftentimes different labels are used because people want to um, earmark these particular study designs as their own when they've been in existence for a long time. Now, exposure. Now we're making a contrast with a case control study. Exposure is determined in the present, okay, and not in the past as in a case control study. And I'll try and make this real clear for you so you get an idea of exactly what I mean. A cohort study conducted by reconstructing data about subjects at a given time or times in the past to evaluate exposure or levels of exposure to a negative health event or disaster. Now, that doesn't mean a lot to you, I know, but we're going to select people in the present, and then we're going to look back to see what they were exposed to, and then what is the current effect on the individual. Okay, if you keep that in mind, you got it. The method uses pre existing records, okay, and other relevant sources to evaluate health status in the past or somebody's vicinity to the disease causing agent or disaster and their current health status. For example, people exposed to the 1917 through 1919 influenza holocaust and their health status at present. And we know that many people that went through the Holocaust, that survived it, had shingles. So we look now at the present, we say, okay, um, who had exposure to the 1917 through 1919 influenza holocaust? Okay, we identify those folks. Then we go back and look at their medical records to verify that they in fact had exposure, had influenza, and then we go to the present and to see what health factors or risk um, were discernible. Okay, so now if we look at just for your information, because I think this information is interesting, the total number of U.S. deaths from this Holocaust was 765,885. And for 1917, areas included 27 states and the District of Columbia. In 1980, 30 states in the District of Columbia. 1919, 33 states and the District of Columbia. Now, I want to give you another example that not it's not very well known, but it's a fact. Um, During the Cold War, and this is true, during the Cold War, the Russians wanted to see whether or not they could survive it. And so they dropped an atomic bomb on one of their own cities. And they wanted to look at survivability over several years and whether there was any abnormalities that occurred, any anomalies in health, and of course, as I mentioned about survivability, how many people live through it. And so, what they did is the president who was involved, and then they went back to check medical records to verify that these individuals were involved in it, and then what were the health consequences in the present. Okay? And they found all kinds of things. They found that infertility, they found parts of the body parts were greatly affected by the nuclear waste. Water was contaminated, and there was a real reduction in the number of people who were alive, but they found that there was a certain portion of the population that could survive this particular trauma no matter what. So I just share that with you as a way to discern and distinguish between a case control study and a historical prospective study. Okay? Now, these studies are done less now because of the growing concern about protection of privacy. But they tell us a lot. For example, I think it's important to know that people in 1917 through 1919 had influenza, and a result of that influenza, they had shingles. So that was a connection that we had noted between influenza and shingles. Okay, that covers really what I want to say about historical prospective studies. And now I'd like to talk about the CADLAC, or the gold standard of all observational studies, which is a prospective longitudinal study. And you can use a prospective longitudinal study if you meet the following specifications. It says when you can associate the disease with a specific exposure based on prior studies. So really what they're referring to is that you might use a more inexpensive study to determine whether you ought to use this very costly study to investigate a problem. So what would be a less expensive study would be a cross-sectional study or a case control study. Okay? You hope that the time is short between exposure and disease. So if somebody's exposed, the ideal is that you would want them to become ill quickly. You don't want something like AIDS, which has, somebody may contract AIDS and it may be 10 years later before the disease is actually present. That takes a lot of time, money, and effort, because with a prospective study, you're going to be having individuals in repeatedly, and you're going to be assessing them probably on an annual basis. And you can think about this. You can begin to think about the problems already in doing a longitudinal study, because follow-up in terms of contact is difficult because people move, people drop out, People get divorced and have other family problems that are more important. They have to fly to a given location, normally to be tested. Um, they stay in a hotel. It's a very difficult and costly study to perform. So the next thing is you have to have substantial funds that are available. Now, we have several of these types of studies going on. One of them is the nurses study, one of them is the NHANES study, and we've had several others, and they provide all kinds of valuable information for us. They're just expensive. Now, it would probably not be wise because of my present age to begin to begin a longitudinal study, because you need to be able to follow people over a prolonged period, because you want to see what diseases develop over time. Now, we did do the Mensa study at Stanford. That's another long-term one, and I don't know what the Mensa requirements are anymore. I thought it was an IQ of 160, but I've heard that 130 is sufficient, so I'm not certain. But you have to have an IQ of a certain number. Um, so we also have that as an example of a longitudinal study. Now, what's interesting about that, and I'm just going to show you this next one so you can, I've already talked about this, but we found out that there was no advantages to having a higher IQ other than being able to learn more quickly. They didn't have fewer social problems, they didn't have fewer anything. They had as many divorces, they had as many financial difficulties, so intelligence wasn't a benefit necessarily in wealth or anything else. I know that at Stanford they would exclude people from the program who had near perfect GRE scores because they had trouble with these individuals in terms of focusing their attention, because they were capable of doing so many different things that they couldn't make a choice as to the one they wanted to focus on. So it became something that was not particularly beneficial, and enhance in an enhancement to their credit or credibility. Okay, so you want to start a longitudinal study if you have a long life expectancy. Now, I want to also make another point here, because I think it's important for you to know, is that what they do when they begin to think about these studies is they have to have somebody waiting in the wings. If the primary investigator dies or loses, or the principal investigator dies or loses interest in the study, they have to have somebody to hand it off to. And as I say, it's a real undertaking that requires a lot of record keeping, but it tells so much about people's health over time and over the years. Now, this study is a prospective study, suffers from the same problems as every one of these studies, in that they have multiple names. A prospective study could be called a cohort study, a longitudinal study, a concurrent study, a follow-up study, an incidence study, and I'll talk about that, and a prevalence study. Okay, now remember, the word cohort is reserved for longitudinal study. Although you saw the word cohort, you're going to say, well, look, I saw that you said with a historical prospective study. If you look at the third bullet, They called it a cohort study conducted by reconstructing data about subjects at a given time. Well, what they're really referring to in that instance is usually when you get to the present. If they want to make it a longitudinal study, then they follow those people from the present through the remainder of their life. So sometimes the word cohort is used with a historical prospective study, but by and large you can count on the fact that this term is reserved for a longitudinal study, and normally people who are well trained in epidemiology reserve this term exclusively for a longitudinal study. Now, let me say this again. This is the only time it's appropriate to use this word unless you have a historical prospective study and then you follow the individuals from the present forward in time. It has to do with following somebody forward in time. So that's what cohort means. I'm going to follow you from this point forward. And it could be on any dimension. I want to make that perfectly clear too. Now, if somebody asked you which two studies are prevalence studies, how are you going to answer that question? Well, we know that the first prevalence study that you became aware of all the observational studies was a cross-sectional study. Now, this can be the first data point. This cross-sectional information that we're going to gather could be the first data point in a prospective study. So this could be the first thing you could do. So can you get prevalence from a prospective study? Yes. Now, what's really unusual is because we're looking at the development of new diseases over time with a prospective study is that you can also obtain incidence. So a prospective study, why it's a Cadillac, why it's the gold standard, is because you can obtain incidence and prevalence rates from this particular type of study. Okay, that's the key to it. That's what makes it exclusive. That's what makes it unusual. And the downside of it is it's so expensive, it takes so long, and you have dropout, you have ethical issues anytime you're dealing with human beings. There's complaints. It's hard to get people to show up for appointments. All kinds of difficulties that go along with it, but it provides rich data. And we've learned so much about people by Ian Haynes and the nurses study, and prospective studies or longitudinal studies. Now I want to show you a couple of common designs. Okay, there are more, but I want to show you the most popular ones, and again remind you that a prospective study can be called by any of these names. If you're studying for a quiz or something of that nature, we will use prospective study. We're not trying to confuse you in terms of terminology. If you understand a prospective study, you understand all of these other terms anyway. Okay, now here's the prospective study, and it mentions that generally there are two types of prospective studies. The first, people initially identified with a known disease and a cohort of unknown diseases are followed forward in time. Okay, so let's just look at this together. Let's take an example. So I have a group of diabetics, and I have a group that does not have diabetes. Okay, what happens to the health of these individuals over time? What's also interesting with the people who are are unknown in terms of their disease, doesn't appear that they have diabetes, is we're wanting to see what the prevalence or the incidence is, because we have a number of new cases, right? When does that occur in their lifetime? How many people, okay, suffer from the disease? What are the health consequences, etc.? Okay. The second one, this is a cohort where the disease status is initially unknown and followed over time. So we have two groups, and they may have some exposure or not, but they are reviewed over time, and they become exposed to something or not, and then they classify themselves into particular study groups, and I'll share that with you in a minute. So don't get confused, just stay with me. Again, I've already made this point, and this is something that you should commit to memory and be knowledgeable about. A prospective study permits the calculations of both incidence and prevalence rates, as well as relative risk based on incidence rates. And relative risk is the gold standard. I haven't talked to you about any of the statistics yet, but oftentimes people will mention to you about relative risk immediately, because we know statistics are associated with study design. When somebody says relative risk, that means a longitudinal or prospective study. If somebody means an odds ratio, that means cross-sectional, case control, or historical prospective study. Okay? So that tells, and sometimes I will see written in the literature, somebody has done a case control study and they report relative risk. Well, I know immediately that's wrong. And there are different ways to calculate relative risk and an odds ratio, and I will go into that. But it's just a signal that they're probably not very scientifically sophisticated, and that's just a real easy way to either ask for clarification or to reject the paper. And usually you find out if they don't know the difference, the quality of the science is usually not very sophisticated. So I want you to know that, again, that statistics go with study designs, okay, and especially with observational study designs. And I will be spending time with this here in a few minutes and hopefully be clear and concise with you as to how we go through the various calculations. Okay. A prospective study design 1. Usually the investigator selects his or her study sample from a defined population, which consists of subjects with and without the disease. Okay? And I want to show you this. And here is how that study design looks. Okay? And let me go over this with you. Okay, so we have a defined population. Exposure status is unknown. And we have a non-random selection. We select from prior knowledge and history and interviews to confirm whether they've been exposed to something or not exposed. Okay? Then what follows, this is time, of course, and we're following them over time. Then we look at the exposed and we see what happens to them. And we know that there'll be a certain portion of those that are, we look what happens to them in terms of their health and health status, health risk. We can look at all the wellness characteristics too, whether if you want to. You could look at physical activity, you can look at mental, emotional health, whatever, okay, spirituality. Okay, now, We know that always that there's a proportion of people who do not develop the disease even though they are exposed. Okay? Now, when we move over to the unexposed group, we're going to have a few of those who develop disease. And we're going to have many that don't develop the disease, and this is what we would expect. So, what we do is we select exposed and unexposed groups, right? That's what it says. Determine at time 2 how many subjects develop the disease in each group and how many do not develop the disease, which provides incidence by subtracting T2 from T1. Okay, so we're looking, over time, how many develop the disease. Okay, and that's what we're trying to do, and we know that if you're exposed, you're going to develop the disease. If you're unexposed, you will develop the disease. Of course, a lot fewer people. And what's made designs like this sort of complicated is, in the beginning, we didn't know anything about secondary smoke. So we would take smokers and look at them, and we had all these people in the supposedly unexposed group because they didn't smoke becoming sick. Okay? So now we have tertiary smokers. And tertiary smokers are those who acquire lung cancer or heart disease. Now remember, with smoking, it's heart disease and then lung cancer are the leading causes of death in that order. And just by sitting in an environment where there's been a lot of smoking and the smoke has been absorbed into the walls and into the furniture, you can develop lung cancer or heart disease. So that was a confounding variable that caused problems, but generally I just want you to understand the design, and I want you to understand the difference between design 1 and design 2, okay, because I'm going to show you another one, and these are the most popular ones, okay? Second design. As mentioned, let me just stay with this one for a few more minutes. It says, as mentioned on the last slide, this is now putting it out in prose, so you can, if I talk too quickly or whatnot, you can read this for yourself. As mentioned on the past slide, start with a study sample drawn from a defined population. Then it says exposure status is known based on prior knowledge, history, interviews to confirm. Observation, screening tests are ways to confirm what you have suspected. And you might, for example, look at a noisy environment if we're checking hearing acuity and what was the difference in those people who were exposed to the noise and those people who were not exposed to the noise. Okay, next, both the outcome variable, hearing acuity, and the risk factors, determinants, are collected at the same time at T1, and that equals prevalence rates. Now, the study sample is followed over time, and at T2, a repeat assessment is done on hearing status, and then we calculate incidence rates. Okay? So, you need a minimum of 2 data collection points to compute incidence rates between the selected exposure and selected outcome variables. So that should give you a pretty good idea about how this study operates and how this is one application, and I use something a little different than the typical health problems that you run into, but you can begin to think about the application to any number of health outcomes that you're interested in. and how a longitudinal study might be utilized. Or when you go to the research literature and you want to know more about a problem, one of the things I would see is how many prospective studies had been done, and I would look at those immediately, because they're going to probably provide you with rich information about the problem. They're going to talk to you about longevity. They're going to talk to you about the incubation period. They're going to talk to you about duration. They're going to talk about all those things in the communicable disease manual. Okay. Hopefully you're on board with study design 1. Now, let's talk about study design 2. Okay? It says exposure is unknown. Currently, everybody is disease free. And we might look at a country, a state, a county, or a city, an ethnic community, an occupational group, any selected respondent characteristics that is of interest to you. And what we're going to do is draw a sample at random. Now, it could be from the Purdue population, for example. And Cohen, and I make you aware of this because this is still the premier book in the area for estimating power statistics, to see whether we have a sufficient sample size to detect a significant difference if one really exists. And people always ask me about a definition of power. And this is probably one of the cleanest, simplest definition of power statistics, or an estimation of sample size. Now, we can't be 100% certain that we have a sufficient number of participants, but generally what I find is Cohen's calculations are pretty accurate and give us an opportunity to really determine whether a significant difference truly exists. And it's not because we don't have enough people that we don't see the effect. Hopefully that makes sense. Now we do an initial assessment to determine that all are disease-free, and over time these people are going to separate themselves into two groups. Okay, and you can see the time period we're representing that over here. So over time something's going to happen to them, and depending on the outcome variable of interest, they're going to either be exposed to something that you're studying, or they're not going to be exposed. Or you may have a wide variety of things that you're looking at, and normally that's the case, and you detect the exposures. It could be environmental, occupational, it could be genetic, any number of factors that you might be looking at in terms of these individuals. And after that, you see who develops the disease and who does not develop disease, and the unexposed is the same way. Okay? So you can see the differences between the two types, and hopefully this is clear and you don't have a problem with it. Now let me present it in a more narrative form for you. And it says, with prospective study 2, you draw a random sample, for example, of college students. You can't test all of them, so but our sampling is what's most important. Remember, I think I mentioned that we only use 1,600 people for the Gallup poll, and we have an accuracy or plus or minus 3%. And the reason why that works so well is the sampling frame of who they're looking for. They pick people who they pick a person that represents many other people just like them. So a fallacy in science is a bigger sample size is better. And that isn't necessarily the case, and it doesn't necessarily rule out bias or error. So anyway, we're going to take a certain percentage, maybe or a certain number, so we would select every 8 student, for example. And we do the power calculations to know how many students we need to select. We would notice this, establish definite criteria in advance for what constitutes a case. For example, the flu and residence hall outbreak. We would go to the communicable disease manual, all those requirements for what constitutes having the flu are laid out there. Serology is mentioned. You know very quickly who's going to qualify and who's not going to qualify as having the flu versus somebody who has a hangover. Okay, because we don't want to include those people as having the flu because they're a totally different group, and we wouldn't want them intermixed, which means confounding, with our group that we've decided are our cases. And so we're going to confirm this through surveys, roommates, and if possible, medical consults to make certain that all are either are disease-free or have the disease. So we're going to sort into exposed or unexposed. Could also institute random sampling at this point if we had too many, or use the matched pairs designed based on demographic factors to reduce or eliminate confounders. Now I'm using terms here that you have not been exposed to. You don't know about a matched pairs design. I understand that. Don't worry about it. You're going to get exposure to that very soon. You don't know about confounders, but I've given you an idea that that's variables that are intermixed that you can't separate, and you can't tell whether A causes B. And that's what we're always trying to do. But you'll learn more about that. And that's why you're taking this class. That's one of the reasons. Okay, let's proceed. And prospective study advantages, management confounding variables, random selection, our matched pairs study, and I'll show you that in a few minutes. Study is not limited by past constraints, allows directionality, but it's not a cause and effect study. It is not a cause and effect study. Okay? The only study that is a cause and effect study is an experiment. It does allow us to say that A comes before B, and that's the first step in determining causality. Then if we wanted to do a causality study, if we wanted to go back to do an experiment to say that A precedes B, we can do so. Now, sometimes what happens in science, because these are all associational studies, all of these observational studies are associational studies that show a connection, a relationship, or a correlation, so we don't know the direction of what influences what, and sometimes we think that A influences B and find out that the sequence is that B influences A, so it's just the opposite. This is why we do science. And as I mentioned, it helps assess causation, but it's still not a causation study, and that is only done through an experiment. And I mentioned that here, and I show it. And one thing about the study that is a real advantage is that there are you don't have to rely on recall because you're going to be doing various assessments, maybe on an annual basis or every other year, and you're going to be collecting the information yourself. Oftentimes it's serological testing. Oftentimes other medical screening procedures are used, such as x-rays and whatnot, and that's why the study costs a lot. But you're really not asking people necessarily, although you can, about a past event. People's memories are foggy, the study sample is going to age, recall is going to become more difficult for some people, they're going to suffer from some diseases like dementia and other factors, okay? And what makes this an important and unique study is that both incidence and prevalence rates can be calculated. Now remember, prevalence rates are calculated by two different study designs. One of the outcomes is from a cross-sectional study, a slice in time, and the other is from a prospective study. And what makes this study really unique is that you calculate incidence rates. Okay, now let's talk about this, the disadvantages of a prospective study. Undoubtedly, they're expensive and time consuming, but our government upon occasion thinks that prospective studies are warranted, and we will learn a lot about health issues, and I tend to agree, and I think they're a very valuable contribution to science, and they're the gold standard. Also mentioned that anytime you deal with people, you can have ethical problems that occur. You can have complaints that run the gamut. These complaints are personnel can call the internal review board that reviews every study and make complaints to them directly. They can be of a discriminatory nature. They can be related to misconduct about how they were treated, any number of issues. The other problem with them is attrition. People drop out. And some of them may die, get divorced, relocate, move to another state or country. You don't have the follow-up information. They don't provide it. And so you lose valuable information from that particular person, and you can't start somebody else because these are a group of people that all start at the same time. So what I'm saying, you can't replace them. And there's a lot more opportunity for bias or error, and we have random or systematic error. Random error is just what it means. If you had a migraine headache, that would be random error because it doesn't occur systematically. You can't predict it. Systematic error has to do with for example, with the poor design of a questionnaire. That's systematic because it's going to occur every time you administer it. Or if you have a phlebotomist that's unskilled in the beginning and becomes more skilled later on, that's a systematic error. Okay, we will cover calculating risks next.